Welcome to The Explainer. I'm your host, Dr. Cam, and today we're embarking on a cinematic laboratory journey right into the incredibly fascinating biochemistry of human digestion. We are going to visualize the microscopic, animate the invisible, and uncover the brilliant molecular machinery operating inside you right this very second. Okay, let's dive right into this. Picture yourself taking a big bite of fresh bread. You know, most people think digestion is all happening down in the stomach, but it actually kicks off the very millisecond that food enters your mouth. We're going to explore the precise biochemical cascade that triggers this incredible transformation.
Here's what we're looking at today. First, we'll look at starch and salivary amylase. Then we'll measure enzyme activity, look at the factors influencing reaction rates, and finally dive into the genetics of human saliva.
All right, part 1, starch and salivary amylase, the biological substrate.
So to really grasp digestion, we've got to understand what we're actually digesting, right? Starch. It's a macromolecule that fuels our cells. Essentially, it's just this massive chain of glucose sugars. But it doesn't just sit there in a straight line. Because of the polarity of the OH groups along the chain, they're pulled toward each other, forming hydrogen bonds. And that causes the whole thing to twist into this beautiful, long, helical coil. It's literally like a microscopic spring just loaded with energy. Now, what's really interesting about this is how our body actually unlocks that energy. Because left alone, starch would take years to decompose into glucose. There's no way we could survive on that. Enter our biological catalyst, salivary amylase. Think of this enzyme as a pair of highly specialized molecular scissors. It zooms in, binds right to the starch chain, and specifically cleaves the alpha-1,4 glycosidic linkages. So it's rapidly chopping that giant helical spring into much shorter carbohydrate units, like the 2-sugar molecule maltose. Moving on to section 2, measuring enzyme activity. Let's take this into the laboratory. To actually see this happening, we use an assay. Now, an assay is basically an investigative lab procedure used to either qualitatively assess or quantitatively measure the presence, amount, or functional activity of a target entity. And for us today, that target entity is the functional activity of our molecular scissors, salivary amylase. Okay, so here's the exact experimental protocol for what we call an iodine assay. Picture our lab bench perfectly set up. We've got 2 test tubes, a control and a test. First, we mix a starch solution, some phosphate buffer to keep the pH nice and stable, and a bit of sodium chloride. Then we place both tubes in a water bath set at exactly 37 degrees Celsius. Sound familiar? Yep, that's normal human body temperature. Then we add a dash of dilute human saliva to our test tube and just plain distilled water to our control. Finally, we terminate the reaction and use an iodine indicator to see exactly what happened. But why iodine, right? Well, it comes down to some absolutely stunning, color-changing chemistry. When starch is dissolved in water, it's completely colorless. But when we add iodine to the mix, a linear triiodide ion forms. This little ion is physically drawn right into that helical coil of the starch. It slides perfectly into the open region in the center, kind of like a hot dog in a bun. When it does this, its electron structure shifts. It absorbs visible red light and reflects shorter waves of blue light. So, an active starch solution suddenly turns this really deep blue-black color. But, and here's the kicker, as our amylase enzyme chops up the starch, that helix gets destroyed. The triiodide ion falls right out, and boom, that blue color completely disappears.
And this brilliantly illustrates an alternative, highly quantitative method we use, called the DNS assay. Now, while iodine shows us the starch disappearing, the DNS assay actually measures the brand new sugars being created. It takes a really precise reagent prep, using a specific mix of 3,5-dinitrosalicylic acid, sodium potassium tartrate, and sodium hydroxide. And honestly, this kind of quantitative assay isn't just for human biology. It's a real workhorse. It's heavily used across food processing industries, biotechnology, and clinical diagnostics to measure enzyme efficiency on a massive industrial scale.
So, when this DNS reagent interacts with the reducing sugars, like that maltose produced by the amylase chopping up the starch, it reacts under heat to form this distinct orange-red compound. We can then take that sample, pop it into a spectrophotometer, and measure the absorbance of light at exactly 540 nanometers. The darker that orange-red color gets, the more sugars have been generated. It gives us hard, undeniable data on exactly how active our enzyme really is.
All right, section 3. Factors influencing reaction rates, thermodynamics and kinetics.
You see, enzymes are incredibly sensitive to their environment. For instance, enzyme activity peaks at an optimum pH, which is right around neutral. Unsurprisingly, that mimics the exact environment of your mouth. But what happens when you swallow that food? Well, it hits your stomach, where the gastric juices have a super harsh acidic pH of around 3.3. At that extreme acidity, the salivary amylase is rapidly inactivated. Its structural shape actually shifts, and well, its job is officially done. And temperature? That's just as critical. Enzyme activity steadily increases as the environment gets warmer. It reaches its absolute peak, its optimal temperature, right around 37 to 40 degrees Celsius, which, again, perfectly matches normal human body temperature.
Let's move to the next concept and see how this builds, because if that temperature keeps rising past 40 degrees, the activity just drops off a cliff. Why? It's due to enzyme denaturation. Basically, the extreme heat causes the protein to literally lose its native 3D shape. And if the active site changes shape, those molecular scissors can no longer fit the starch chains. The lock and key are completely broken. Beyond just temperature and pH, we also have to consider substrate concentration, which is governed by Michaelis-Menten kinetics. The idea here is that if you add more starch, the enzymes will work faster, sure, but only up to a point. Imagine a microscopic factory line. You can keep speeding up the conveyor belt, but eventually every single worker, or in this case, active enzyme site, is completely occupied. Once all those sites are saturated, you hit a maximum rate known as Vmax. At that point, the factory simply cannot process the starch any faster.
And finally, Section 4, Genetics of Human Saliva, the Individual Blueprint. You know, salivary amylase isn't just some generic chemical floating around. It's heavily dictated by your own DNA, specifically the AM1 gene family, located on chromosome 1p21.1. Incredibly, this one enzyme makes up 10 to 20% of the total protein in your saliva. But here is where it gets super fascinating. We don't all have the same amount. The copy numbers of this gene actually vary drastically between different ethnic groups. Studies show that populations with a historically high carbohydrate diet have literally evolved to have higher copy numbers of this gene. That means they produce way more amylase in their saliva to handle that specific dietary load. It's just amazing. So the crucial point is the molecular mechanics of how this gene actually translates into physical structure. The enzyme features this really cool glycine-rich flexible loop that acts kind of like a physical gate. When there's no substrate, it's open. When starch binds, it clamps down to hold it in place for hydrolysis, and then it opens back up to release the maltose. Furthermore, this is a calcium metalloenzyme, meaning it relies absolutely on a single calcium ion to stabilize its structure. Without that ion, this brilliant molecular machine simply cannot function. Which really leaves us with a pretty profound thought. Your saliva is actually a highly customized biological fluid sculpted over generations by the diets of your ancestors. So the next time you take a bite of bread, just think about those molecular scissors, the complex thermodynamics, and the deep genetic history going to work in milliseconds inside your own mouth. It makes you wonder, how else might our modern diets be shaping our genetic blueprints right now? Definitely something to chew on. I'm Dr. Cam. Thank you so much for joining me on this explainer. And keep exploring the science inside you.